All right, so now you have your alignments. BWA did the alignments. Picard sorted and removed the duplicates. And so now we want to go back in and actually use GATK's tool set. And so base recalibration is their method. And so they go back and look at the scores, the quality scores that were produced by the machine, and look to find out what type of technical errors might have occurred. And so this base quality score recalibration, or BQSR, according to GATK, is a machine learning that models those errors empirically and adjusts the quality scores accordingly. And so something that might have had a really good quality score but was due mostly to machine sequencing errors, this BQSR tool will come back and assign the appropriate quality score for that. And so the accuracy now of those bases with the Q scores get much better for downstream variant calling. And so here, base recalibration is a two-step process. You first must run base recalibrator on each of your samples. And so here I'm going to, you'll see this is a lot of code to type out. I'm going to copy and paste it. To quickly cover what we just copied and pasted, we're using GATK here, and so it is also a jar file, so you need to run Java jar, and then you point it to the location of where you have the GATK jar file. In my instance, I have it in a directory called tools. The -T is the tool that you'll be using inside GATK, and in this instance we're using base recalibrator. The -R is different from the BWA-R that you used a while back, so I encourage you to definitely make sure you understand all your options and parameters. But this is pointing to the FASTA file, so that you aligned your BWA samples to. So the -R, you point it to the directory and then to the FASTA file that you aligned your original samples to. The -I is your input. And so we're going to input the father_dedupe BAM or whatever sample that you need to run. The -L is very helpful if you have exome data or targeted sequencing. In this instance, we're only looking at a very, very small portion of chromosome 20. This just gives it the region to look at. It helps run GATK a little bit faster. If you're doing whole genome, you won't need this option because you're looking at every single base. Then you provide it the pedigree file, which lists the father, mother, and son and their relationships and who's affected and who's not. Then you provide various known sites. So for dbSNP, the Mills and 1000 genome gold standard, along with 1000 genome phase 1 data to help better make these calls for recalibrating the quality score. And you can see my various commands or my options explained here. All right, so the father ran fairly quickly. Let's run through the mother sample. And I'm going to go in and start to copy the sons. And we'll paste the sons here. Now we've done our first step in the base recalibration. All right, so we need to now run a second pass using base recalibrator from the output file that was created in the first round. And there will be one extra option parameter that you'll need to add. The command looks very similar to what you were just copying and pasting, or if you were typing it out, it's typed out here. But the one extra option is we are providing this BQSR option parameter, and this file was generated in the first pass here. So we created that particular file when we ran this command. Basically what we're doing is a two-step process. We want to see what did the quality look like before and what did the quality look like after it got cleaned up with the recalibration step. And so these create PDF files that you can view to see the before and after effects. So let's copy and paste this line. Basically, again, it's just the same thing, but we've added in that line and we've are changing. So we outputted the initial data group file in the previous step, and so we're now feeding that into our now second pass here, and now we're outputting now this data group file, this final data group file. So you hit enter. And we'll go ahead and copy the mother because these will take not very long. Here is the father, or no, the father was just run. Now I'm running the mother. Now the son. And so now those two GRP files, the group files basically from the base recalibrator first and second pass, have been created. And so we now want to take those two files and run the analyze covariate step. And here you will provide it the before file, which I had labeled the initial _ data group, and then that was the first pass of the base recalibrator. And then you give it the after file, which was the second pass of the base recalibrator step, so it had the final _ data.group. Here I will have it create plots, and it will create a PDF file, so you can give the name of the file however you want to name it. I even tell it to create a CSV file that has all the actual numbers. If I don't like the way it looks in the PDF, I can always go back and create my own plots. So copy and paste. And you'll notice that I'm now doing a lot more copy and pasting because these GATK tools, options and parameters, are much more longer. There's a whole lot more to type. I definitely have my own scripts for all of these, so I encourage you to build your own scripts to avoid typing, which in the end creates typos, and then a lot of extra troubleshooting. And so these will create the PDF files and the CSV files that so that you can look at the before and after effects of how this base quality score recalibration step worked. Okay, great. Here are the options that I used, but again, there are many more that I did not cover. So, you know, be sure to understand them, understand why I used the ones I used, and if you need to use other ones, make sure you put those in at the time. All right, so now that we have the files that tell us how to go back and change the base quality scores, we now need to do a step called print reads. It's a very simple command calling in the GATK jar file. We're calling in the tool print reads. We're giving it the FASTA file of the reference genome. We're inputting in the dedupe BAM file. We're giving the location of the region, again good for exomes, targeted sequencing and such. Providing the pedigree file. I'm giving it the BQSR initial first file that we did in the base recalibration step first pass. And then I'm outputting a brand new BAM file, which I call whatever the name of your sample. In this instance, it's father_recal, short for recalibration.bam. Again, you name it however you want to name these files. So copy and paste. And so this is now taking those base quality score recalibration results and implementing it now in the new dedupe BAM file or in the new recal BAM file that we just generated. Copying and pasting the mother sample. And copying, and we'll soon paste the sun sample. Again, at the end of every one of my steps, I give you a brief explanation of the options parameters that I used, so feel free to look over those. All right, we are now done with phase one. We have pre-processed our FASTQ files all the way to now a recalibrated alignment file. So good job. Give yourself a pat on the back for making it through this. You are 1/3 of the way done with the calling variants. So we're now moving into phase two, which is the variant discovery step. We are going to provide the recal-BAM files that we just created in the last step into the haplotype caller, which is going to do our variant calling. It's going to call the SNPs and indels. We will then, and you do this for every single sample, we will then join everything together into one VCF file. So now, so you would run this step here, the haplotype caller, on all your samples separately. So in my instance, I have 3, so father, mother, and son. Run them this way. Then you joint genotype and bring them all together. So now that you have one file that has all of the samples together in one, and so you have this raw list of variants, much like what we did with the base quality score recalibration. We're going to do a variant quality score recalibration step to clean that up, and we'll have analysis ready variants at that point. So let's start with the beginning. Let's call our variants. Let's actually now make a VCF file, a variant call format file, that lists all the variants. And so we're going to use haplotype caller. The GATK has another caller called Unified Genotyper. Haplotype's a little better, in my opinion, but I will encourage you to go look at their website and read up on the differences between the two and make your judgment call yourself. All right, so we need to run the haplotype caller. We're going to call variants on each of our samples. So again, you run this command 3 different times. In my instance, depending on how many samples you have, will dictate how many times you need to run that. Copy and pasting. So here I'm running GATK, providing the path to GATK. The tool I'm using inside GATK is called haplotype caller. The -R option, I'm providing the FASTA file that I used as my alignment. My -I is my recalibrated BAM file that I had just created in the previous step. The -L is the location that I'm most interested in this instance is this very small region on chromosome 20. My genotyping mode is set to discovery. I've got the minimum Phred scale confidence threshold at which variants should be called, so I've got it set to 10. I want to output a VCF file, so I'm giving it the name of the sample, father, and this is going to be using haplotype caller, and this is going to be a genomic VCF file, providing my pedigree file. I wanted to output the genomic VCF file, so you have to provide that option. I'm giving a dbSNP database of 138 in just a particular region, so that way it will mark inside there if it has been seen in that particular version of the dbSNP database. All right, so that ran for the father. Now let's do the mother. So we'll copy and paste the mother's command. All right, the mother's sample has been processed. We have a variant file for that. Let's copy the son's. And paste it, and we'll run that analysis on the haplotype caller. Let's see here. We should see a VCF file here for each of the samples. So father will be one, then there will be one for the mother and one for the father as well. All right, so now we have a raw set of variants that have been called. And now we're ready to go to the next step, this joint genotyping step. And so here, this will merge all three of the VCF files that we just created into one file. So one command line to type, yay, no more, one for every single sample. This is where we're now merging everything together. So we're going to copy and paste this command. Hit enter. Again, you point it to the GATK jar file. You're giving the tool set genotype GVCFs, the FASTA file, your pedigree file, the variant files that you just created. So in my instance, I have 3 times I'm calling variant, and after each time I list variant, I list the father, or I list the VCF file. So I have my son, I have the father, and I have the mother's VCF file. If you have more, you just keep typing variant with the name of the VCF file that you've created. I then will create an output file. So now I'm just calling it family. .raw.snips.indels.vcf. Again, that can be modified to whatever you want to name your samples. And then I'm giving it the dbSNP database again. We now have one file, which you should see here, this family SNPs indels, that now contains all 3 of the variants, all 3 of the samples variants called in one file. Makes life a little bit easier from now on. All right, so much like what we did with the alignment files, we need to go back and do a quality score recalibration. This time, instead of doing the base quality, we're now doing the variant quality score recalibration, or what they call as VQSR. Does similar things as the previous one, and so it's very helpful to clean that up. Now, this is where I will diverge slightly from the GATK best practices pipeline because this step is so small. Not this step. These samples has got such a small region. We're only looking at a very small region on chromosome 20 that this step will fail. And so if you have an exome data set, or if you have a genome, it shouldn't happen with genomes, you can come in, if this step fails, you can come in and apply hard filters. And go look up on their best practices guidelines of the types of hard filters you can put into place. However, if you are doing a real data set, I did provide the commands here on how to process this. Instead of doing hard filters, if you can run this. So I wanted to provide all the parameters here and the options I use as a default. Again, modify them accordingly. So here we're going to have to do the variant recalibration and a apply recalibration step on these on the joint genotyped file. And the first step, you run it on the SNPs. So here you're calling Java, you're providing the path to your GATK tool, you're providing the tool that is inside GATK, so we're calling variant recalibrator. Here's the FASTA file. We're inputting in the VCF file that we just created from the joint genotype tool step. And then you provide it various resources, the HapMap, the Omni, the 1000 genome, so forth. The GATK pipeline has a table that lists when to use which resources and what settings to use. Here I'm just running mostly as is what they say in their default, but there are times where I've had to modify these, so be aware. So these various resource parameters need to be modified accordingly. I'm providing the pedigree file, and then various markers like depth, Fisher strand, strand bias, mapping quality, and so forth. I want to put in my info tab column. Here I'm telling I'm only wanting to focus in on the SNPs. I'm setting up my tranches, or my bins as I like to call them sometime, as to where things will fall. This can also be modified. I'm leaving it as their default way, but I have had to modify these before. I'm producing a recalibrated file that just basically, much like the base recalibration step, this will create a variant recalibration file and a tranches file and an ARS file, so that you can come back and plot these later to see the before and after effects. So once you have that run, that is all one command here to do the variant recalibration step on just the SNPs alone. Once you are finished with that, you then need to apply the recalibration that you just created here. And so you're going to provide this file that was generated down here. All right. So here again, we will call Java jar the location. The tool is apply recalibration. The genome that we're using by the -R option. We're inputting in the same VCF file that we just had in the previous step. The mode is still SNP. The filter level needs to be set to the what you need to set it as. Here I've got it at 99. Here I'm giving it the recal file that was generated up here in the previous step, and the tranches file that was also created in the previous step, the pedigree file, and then I'm outputting a new VCF file. So I gave it my raw SNPs indels file, and now I'm coming back and outputting a brand new variant file that I've labeled family.snips.recalibrated.indels.raw. So this just tells me that my SNPs have been recalibrated, the indels have not, because we're about to touch the indels here now in the next step. And so we now need to go back and run variant recalibrator again, now on the indels. The two commands prior here, we ran it on the SNPs. Now we need to run it on indels. So here we're providing the path to GATK. We're calling in the tool variant recalibrator. We have the -R option, which points to the reference genome. We are inputting in the VCF file that we created in the previous step here. We now provide a different set of resources because this is indels. So take a look at their table on GATK's website. Here's the pedigree file. Again, the various settings, the various filter parameters are the things that I want to see inside the info. Here, instead of mode SNP, we now change this to mode indel. Modify your tranches accordingly. And this max Gaussians can be used as well. And here we're going to create a recalibrated text delimited file on the variants named family.indels.recal. We're going to create a tranche file and another R script to create those plots for downstream to see the before after effects. All right, now you've got your recalibrated file, your tranches file, and now you need to apply the recalibration on those indels. So here we take the Java file, the Java jar file, and the GATK path. We're using apply recalibration. Here is the reference genome. We're inputting in the SNPs recalibrated indels raw variant file. We've changed the mode from SNP to indel. You modify your TS filter level accordingly. You give this tool the indels recal that you had created in the previous step. The tranches file, same in the previous step, you take that file, provide the pedigree file, and you output a brand new file that now has both the SNPs and indels. They've both have now been recalibrated. And so at the end of that step, you have just finished the phase 2 of variant calling discovery. So you've now 2/3 of the way done. Now, none of this was done at command line because, again, as I mentioned, this sample was such a small region, but you shouldn't have a problem if you have several samples that have been done in exome sequencing or whole genome. So run those 4 sets of commands there that I just walked you through, and you should, at the end of the day, have some sort of file that I've got here highlighted that I've labeled it this way, but however you labeled yours. So this would be the actual file that you would then take into the last phase of call set refinement.